Hi, Katie. Thank you for responding to my initial post and for the kind words you gave me. I really liked your arguments as well. I think that the point you made about how not the prompt, but like how design, incentive, and culture, it's a bit simplified of a view of the issue because it really is such a multifaceted issue. Uh, Reproducibility, I mean. But yeah, I really like this approach you had, and I like the idea that culture is the driving sort of force. Although I think I slightly disagree, not even in anything you said, but just in the naming. I think that it is a cultural issue, but I think that that culture is pushed by incentives. But to be honest, it's kind of like a chicken or egg situation. Like I'm not sure if it's incentives which push it or if it's the culture, because you referenced that it's publish or perish, and I agree, but I'm not sure if that's an incentive thing first or a cultural thing first. I don't know if that makes much sense, but that's kind of how I feel. I think there is an overall like incentive push from academia in general to kind of have um, a high volume of papers, positive results, and stuff like that. And that is that incentive works because that is where they draw funding from. Academic institutions usually, well, not to be too general, but like often they can be nonprofit, which means they pull from grants, and sometimes they can seek like private investing for grants. And if it's private, if they're looking to find private funding, it's often because they're publishing results that that private institution agrees with or that they are interested in exploring more. So that's kind of why I believe it's an incentive issue. Of course, like the cultural thing is like hand in hand with that because you have obviously scientists who are successful in their field because they've followed like a certain paradigm, and then they will then transfer that to, their mentees. um, so yeah, I think it's a really complex issue, and I really like your approach to it. I think the part where you kind of referenced a paper which was or a study which was very standardized, and the experimental design was top tier premium, and yet they still found variability. I think that speaks more so to how complex science is, especially in biotechnology, more so than experimental design, because you can design everything perfectly, you can do everything as standard as possible, but at the end of the day you may still have some variability just because we are studying such a complex system, basically. So yeah, I think it's a big issue. um, Experimental design, you can do everything right, and at the end of the day, you may have, reproduced data sets that don't agree with each other. That's kind of why I believe that, like, although we should rerun all our analysis and statistical tests and reproduce the same result or reproduce the study with the same data, I also think it's beneficial to start from scratch and recollect as much data as you can to then prove a previous finding with a completely new set of acquired data. Um, I think that just kind of adds a layer of reproducibility, and it would definitely help uh, either corroborate or contradict findings, I think. Um, it's also really important to, yeah, as you said, it's very important that instead of just publishing methods, like a methods paper, it should be like in person or at least like direct communication between labs and scientists in order to figure out how they approach the experimental design, exactly how they did their analysis, how they did their data collection, even the way they set up the lab. I believe it's like a bit interesting to kind of want to recreate that as much as possible because different tools obviously have slightly different accuracies, different, environments have different contaminants. It's a whole thing, right? So how much can we really standardize without literally doing it in the exact same room? And then at that point, is it due to like being in the same room, or So yeah, it's really interesting. I like your opinion that culture is the hierarchy issue, and then everything else sort of falls below that. Because, to be honest, how do you rectify experimental design to the point where you can make it hyper reproducible without first addressing culture? So I really appreciated your argument there. I also liked how you said that reproducibility can be socially uncomfortable, um, and that like it's a bit there's a bit of tension between scientists that disagree with each other, and how do you rectify that as well? Um, I think that definitely is cultural over incentive or anything else because it's like it's part of the business. You simply have to publish the results even if they contradict the previous findings. It's just what you have to do. It's not a personal thing, right? It's part of a larger kind of effort. So I like that. I like that insight into like kind of the mind of a scientist. Like, let's say you get a result that does not agree, and you publish it, and you kind of don't want to publish it because you don't want to hurt the credibility of another of a colleague, right? But you have to understand that that is the purpose of reproducibility. You must do that. And yeah, I really liked that kind of insight you had. I thought it was really powerful. Um, I think I already touched upon how I'm not too sure it's very easy to separate culture and incentives. I think we're in agreement of this, just phrasing it a bit differently. You believe that the prompt is slightly oversimplified and that it's too much of a combination of the three. I also agree. I don't think it's very easy to separate culture and incentives. Um, yeah. Overall, I think he had strong points. I really liked the kind of insight into the psyche and like social thinking of scientists. That I think that's pretty powerful to consider it from their point of view. I'm not too sure about the culture versus incentives thing. It'd be interesting to discuss further. And yeah, thank you.